Hello. Really quick 8D, pedigrees, mutations, and karyotypes. Pedigree is the way we can actually track parental makeup of generations. We show females this way, males this way. Females round and males square. This is a girl without a trait. This is a boy without a trait. This is a girl with a trait. We just fill it in. And a boy with a trait. We fill it in. If we're going to do carriers, we fill in half the box, which means the trait's not showing up, but the trait can be passed on to the next generation. Here we're showing a mom and a dad, and we're showing a direct relationship between the son and the daughter, all of which are without a trait. And this is a pedigree, and we can follow this from grandparents, great-grandparents, go back as far as the ancestral tree makes a record. That's the wife and the husband, and there are the children. What can you tell from looking at a pedigree? You can tell the number of generations, in other words, the number of lines that are in there. You can tell relationships in terms of marriages, brothers and sisters. You can tell dominant or recessive, whether it shows up in both sexes or whether it shows up in one sex. This usually shows up in all generations. This usually can skip one or two generations, but usually not as prevalent as the dominant. We can show sex length and autosomal. Sex lengths usually affect one particular sex. Usually it's a male, autosomal, any sex. So here we actually have carriers, and if you take a look down at the key, we have homozygous male. This is a male that actually is fine. I'm sorry, this is because we have wild typed up here, this is probably fruit flies. This is a male that actually has the trait. There's a female with a carrier, there's a male carrier, another male carrier, another male carrier, another male carrier, another female carrier. So this is telling us it's not sex trait, it's an autosomal because it's in every sex. It also is showing up in, actually it's only showing up in one generation there, so this may be a recessive trait. Definitely is a recessive since the heterozygous is not showing. So we have 1, 2, 3 generations. We can actually see husband or wife had one daughter, one son, two daughters, two sons. They got married. This particular female was a wild type, picked up the blue from the dad and the blue from the mom, married a carrier male. We actually have a male blue, married blue. We have a female that did not get married, and we had a male that actually has the whatever this trait is, married a wild type female, or mated with, since this is probably, again, fruit flies. And then we can see 3 kids, 2 kids, 4 kids. We have 2 carriers, nothing, and 1 carrier male, one of each here. Dominant or recessive? What do you think? Showing up only one generation, I would say it's recessive. Is it sex-linked or autosomal? Well, it's showing up in both sexes, so it's an autosomal. How many generations? 3. Here's another one. We have a husband and wife, had 1, 2, 3, 4 kids. Looks like it actually is showing up in all sex. Is it dominant or recessive? Showing up in every generation, so that makes it dominant. Is it sex-linked or autosomal? Looks like it's actually autosomal. How many generations? We have 3 again. Okay, here we have another sign. This sign is actually showing that they're deceased. So we actually have everybody deceased in those 2 generations, and we have 1 deceased here. So we actually have nothing showing up there, and I'm not sure exactly what goes on there, but it looks like it's actually showing up in some generations, not all, so it's a recessive. It's showing up in only males, so it's sex-linked, and again we have 3 generations. Here's another one. If we take a look at it, it looks like it's showing up in males and females. I'm going to say that one's dominant, and we're showing up males, females, so it's autosomal, and we have 1, 2, 3, 4, 5 generations in this one. Then we have karyotypes. The way we can make a karyotype is we get a cell, and we get it in pro-metaphase, halfway between prophase and metaphase, where the chromosomes have formed. Chromosomes are setting up, but they're not quite in the center. And then what we do is we smash the cell. Basically what we do is we break the membranes open. We pull out the chromosomes, and then we try to line them up showing like chromosomes, and here we actually have, looks like we have 23 and 23. We have both male and female. Actually, what we've got is we've got 23. Well, we've got the chromosomes, which we're going to stick over here. It's probably better to show it over here. And what we have is chromosomes that actually have the centromere sort of in the middle, where we have symmetric chromosomes. And then we have some that actually put the centromere up toward the top. We have asymmetric chromosomes, and you can see we get smaller as we go down, and then the 23rd pair, the X and Y, this would be a male. Pictures on, I'm actually going to pause this because I got a lot of noise. I'm at school. This is the day after St. Patty's Day. We had a snow day yesterday, our 13th of the year, and we have our 9th 2-hour delay today, and teachers are showing up. It's like 8:15 in the morning. Kids don't show up for another 45. But anyway, we've got the chromosomes in here. This would be a male. Karyotype is a picture of the homologous chromosomes all lined up so that they actually are the matching pairs, and it's amazing how somebody can go through here and do this. I put a website up for my snow day assignment for my biology kids, allowing them to actually see one of the chromosomes and then a hodgepodge of other things, and they were supposed to drag them over to see how easy it is, which it's not. Then we have what's called non-disjunction. When we actually break these chromosomes apart, sometimes we actually get them so that they actually end up with N plus 1. This would be a normal one. We actually have an extra chromosome in there, so we get a zygote that has the 2N, but we actually have 2 of the red ones from, looks like mom, and this is a non-disjunction, which means that the chromosomes didn't break up correctly. When chromosomes do not separate correctly in meiosis, and you can see a non-disjunction. We go there, and we have 3 in this one and 1 in this one, not good. And then we have a normal one where everybody looks like they're the same, except that we actually get a non-disjunction right there. So we get a non-disjunction in meiosis 1, this is the non-disjunction in meiosis 2, and we have asymmetric things. So if this was actually the egg in oogenesis, if this was the one that stays, these would become polar bodies that would not show up. If this was spermogenesis, making sperm, we would have a problem because we'd have 2 that don't have enough and we have 2 that have too many. If a mutation happens in a somatic cell, the person with the mutation is the only one that's affected because it stays in their body cells. It doesn't get passed on. So there's a normal cell, here's the cancer cell. If it happens in a somatic cell like the liver, you're not going to pass that on to your offspring. What if it happens in a germ cell? Then you're going to pass it on to the next generation, and that's non-disjunction. So if this leads to a genetic disorder, congratulations, you have a bouncing baby boy or girl with that genetic disorder. Here's the karyotype. Is this thing a male or a female? All we do is look down here at this number 23. It looks like we got an X and a Y. This is a male. Do you see anything that looks absolutely weird? And I'm not talking about these shapes, because when we actually do this squishing, the shapes can change on the chromosomes, but this is a no. This is a normal male. Here's another one. Take a look at it. Looks like we got a female, and if we look at them, we got a non-disjunction. We have a trisomy 21, and that's Down syndrome. Here we have what's that? We have a male and a female. But we call it a male because it's got the Y. It's got 2 X's and a Y, which is going to make what's called a super male. Then we go to pedigrees, mutations, and karyotypes. The last thing we have to talk about in 8D, the type of chromosomal mutations. We have a deletion where we actually take out nucleotides, and you can see we've lost the CD in this one, shortening it out. This is going to change the protein again, which may or may not be a problem. Here's an insertion where we're going to put another CD in, so now we actually have an ABC, where we're supposed to have ABC, D, E, F. We have an ABC, D, C, D, E, F, G. So we actually have a different coding for protons, or for proteins. And this is when a segment is deleted or lost. This is when we add it or insert it. Then we have what's called an inversion, where the CDE flip. And instead of an ABC, we have an ABE. Instead of a DEF, we have a DCF. Okay, and that could again change the protons. And then translocation, this is where they actually end up in different places. So we actually have this chromosome and this chromosome, and we actually get this section right here and that section right there to flip. And you can see the AB of the gray, EFG of the yellow, and ABCD of the yellow, and an EFG. So this is when a segment is flipped or reversed, and this is when a segment of one chromosome gets swapped with another chromosome. And if you take a look in there, we actually have some pieces missing out of this chromosome 1. We have it in chromosome 3, and that may or may not cause a big problem. That's it. Thank you for stopping by. Appreciate it.